or epigenomics came in. What's that all about? Is that something to do with the way that genes during development inherited from mum and dad interact with the environment? Well, luckily we have the answer to that question because we have a stellar epigenomics group in the Institute, and it's a pleasure then to ask them to come and present to you. And I think Professor Karen Lillicrop is going to kick that particular ball off. Karen.
How to do this. Okay, so today, Chris Bell, myself, and Keith Godfrey are going to tell you a little bit about the work that's been carried out in the IDS over the past 10 years, looking at the role of epigenetic processes in the development of human disease. So traditionally, it's been generally accepted that our genes determine our phenotype and susceptibility to disease. But is this really the whole story? Are genes really the sole determinant of disease risk? Because there have now been many studies in twins which have shown that twins are often discordant for a wide variety of disease types. So often you find one twin will become obese, the other will stay lean. One twin will develop diabetes, the other will not. Yet they share the same DNA. We're also beginning to learn that not all cloned animals are the same. And probably the most famous example of this is Rainbow the cat. So the history behind this cat is that a group of scientists decided to produce the first cloned cat. So they decided to call Rainbow's clone Carbon Copy, expecting Carbon Copy to be an exact replica of Rainbow. But this is what Carbon Copy looks like, and as you can see, very different indeed. Yet again, these cats share the same DNA, the same sequences of bases within their DNA. Also, these two mice are genetically identical, yet one is yellow and obese, the other is brown and lean. So how do we explain then this difference in phenotype and disease risk in these genetically identical mice? Well, we're beginning to learn that above the genetic code lies a second code, and this is the epigenetic code, which integrates information from the environment and alters the activity of our genes. So in fact, phenotype, disease risk, is determined by a combination of our genes and these epigenetic marks. So these processes which control the activity of our genes are able to actually control the expression of the genes without altering the actual sequence of the DNA. And many people have likened genes and epigenetics to a computer, with the genes being the hardware, the epigenetics the software. And the three main epigenetic processes are DNA methylation, histone modification, and non-coding RNAs. And today we're going to talk about the methylation of DNA, as this is where most of the work in the IDS has been focused on. So when DNA is methylated, it is primarily the cytosine base that is methylated. Most of the methylated cytosines are found next to a G base. So people tend to talk about CGs or CPGs. And in general, low levels of methylation mean that transcription factors can bind to the promoter region or control region of a gene, and they can activate that gene. So you get a production of an mRNA and a protein. In contrast, high levels of methylation prevent those proteins from binding to the control region of a gene, and so the gene remains inactive, so you get no mRNA, no protein being made, and the gene stays switched off. But what we're beginning to learn is that you can get alterations in these methylation marks on our DNA, and this then leads to a variety of human diseases. And Chris is going to tell you a little bit about that now.
Great. Okay, so I'm going to give you a quick fly-through work that I'm involved in using large-scale data sets to try and understand common human disease. So you're obviously aware that whichever environment you place yourself in, a healthy environment or an unhealthy environment, it's going to have a dramatic risk on your likelihood of developing a lot of these common diseases that we develop as we age. So the reason why we want to look at the epigenome is because of the fact that it's tissue specific. So essentially it gives us a window into the molecular workings of particular cell types and how those may be abrogated in disease. So when we're analysing the epigenome, we can either do this to look at its function, so how we can see changes in gene activity in particular cell types that can inform us about disease pathophysiology. But there's also another side to looking at the epigenome. We can actually use it as a biomarker for exposure. And this is fantastic for epidemiological studies because we can get measures by looking at samples of DNA that we've had sitting in the fridge for years about how much that individual smoked by changes in their DNA methylation. We can even see changes in individuals whose mothers smoked. We can see the effect of passive smoking and even ex-smoking. And what's really interesting is, and we can actually use these changes in DNA methylation to measure what we're describing as biological age. So these changes in DNA methylation are so accurate that we've been able to formulate an epigenetic clock from Steve Horvath and others. And what we can see is that individuals that we predict are actually a lot older than their actual chronological age. So when they have age acceleration, biological age acceleration, are at a huge risk for a lot of diseases that have an increased risk of both morbidity and mortality in large epidemiological studies. So when we're studying the epigenome, what we're talking about is we're looking at a particular mark across the genome in a multiple of different individuals, and we're trying to use that information to tell us about disease, as I say, about function, or we can utilize this information as an exposure biomarker. But we also need to be aware that the genetic background will also influence this epigenome that we're assaying. So there may be common variation in, for instance, the binding site of a transcription factor that influence the likelihood of that happening, that then will influence a regional function, a functional regional region, such as an enhancer. So firstly, I just want to describe a couple of collaborations that I've been involved in that are really utilizing the tissue-specific nature of the epigenome to understand disease. Firstly, a collaboration with Sandrine Geraton at UCL, and she studies chronic pain in a rat model. So what she does is she dissects out the spinal cord dorsal horn, and what we've been able to identify is there are specific DNA methylation changes in this FKBP51 gene that are also associated with gene expression changes. This gene is involved in glucocorticoid signaling, which is related to this chronic pain, and this can potentially open up novel therapeutic avenues for chronic pain. The common disease of type 2 diabetes, here a collaboration with Mark McCarthy and Anna Glowen at Oxford. So what they've done is they've devised a pancreatic islet regulome. So they've looked specifically at the pancreatic islets. They've combined some whole genome bisulfite. So this is looking at the DNA methylation across the whole genome, as well as some ATAC-seq, which looks at openness or ability to be regulatory, as well as some existing chromatin data. And what they've found is regions that have high activity are strongly enriched for transcription factors that we know are important in the function of the pancreatic islets. And you can use this information to start to understand the genetic susceptibilities that we've identified through GWAS in type 2 diabetes. So now I'm just going to describe, and hopefully don't get lost in the complexity here, but simply what I'm trying to do here is we've now got some really large-scale studies where we can actually look how the genetic background is influencing the epigenome that we haven't really been able to do with array studies because we're just looking at individual CPGs with this. This is sequencing-based studies, and we can really start to see the impact of large-scale genetic variants on what we're measuring in terms of epigenetic change, and how this may confound studies, but that also may give insight to function. So what we did a couple of years ago, we took all of the GWAS catalogue, so all of these common variants that have been associated with common disease, we identified their linkage disequilibrium blocks. So this is the region that's sort of co-inherited with the association. And we looked at the DNA methylation across this in 3,000 individuals in peripheral blood. And essentially then we categorized the individuals by whether or not, how many risk alleles for the GWAS SNP that they carried. So essentially what we're looking for here is if there's any linear relationship between the methylation in this region we know that genetically is associated with a disease. And what we did is we identified these robust haplotype-specific methylation peaks within these regions that are associated with all these traits. So, what does this mean? Well, as I mentioned, we were concerned that large-scale genetic variants might be influencing this. And we do see this to a certain degree. So you can see this is the linkage disequilibrium blocks themselves, and this is the peaks. You can see there's a big enrichment for copy number variation, and these other large-scale variants like microsatellites and indels, but they don't take up all the signal. So we can count those, but we can also see that there's a lot of additional evidence that these regions are functional. About 10% of them overlap enhancers. And there's some interesting other functional information that tells us that we've got allele-specific differences in their function that may be related to these common diseases. So as I say, this is taking epigenetic data and trying to understand the genetic variability that we know is associated with a disease. What we also did in these regions, in fact, was to look for aging changes as well. So again, just to mention this is in peripheral blood, we found a much smaller number of aging changes, just 71. We started off with a discovery set of 2,000, and these strongly replicated. But where we found these aging changes, they were strongly enriched for enhancers. So we can see almost 50% overlap with enhancers, more than expected. What was really interesting as well is that where we saw these aging changes, they were more likely to be in GWAS regions or diseases that we know have an aging-related component. So as I mentioned, these findings were in blood, so they're really interesting for the GWAS findings for blood-related diseases because there's direct tissue-specific variation that we can integrate. But we're also interested, are these changes important in the other diseases as well in these GWAS regions? So what we did is we took a general indicator of activity, this is DNA's hypersensitivity sites, and we overlapped it with a huge range of different tissues. So the length of the bar is the enrichment for overlap. So we'd expect to see a strong enrichment in blood tissue, that's what we're looking at, and these are all the blood cell types up here in red. But you can see it doesn't fall off in the other tissues as well. So potentially these changes are occurring in regions that might be interesting for these other tissue types, but we'd really want to investigate those specifically themselves to confirm this. Yeah. So, and some interesting biology that came out of this. One of our top hits for hypermethylation was over the locus of the tRNA initiator methionine, so obviously the required first amino acid. So we were quite curious about this. What does this mean in terms of aging biology? We know that there's a large number of copies of this. There's variation of this across tissue. So we set out exploring this, and this is work that my PhD student Richard Acton's doing. There's a poster outside that you can have a look at. And essentially he's looked at approximately the 600 tRNA genes in the human genome and shown that they strongly hypermethylate with age. And he's trying to unpick what that biology may mean. So just to conclude, essentially what we're doing is we're integrating all of this epigenetic data we have, pure data about particular tissues, exposures, age, and also genetic background, with what we know about genetic susceptibility. We've done all of these huge studies now, and we know a lot about common variation related to disease, but we don't know how to understand the function. So by integrating this together, we can potentially identify function, and hopefully that leads to new treatments or prevention. So just to conclude, it's my pleasure just to present a couple of slides from the imprinting group. So this is led by two world leaders we have here in Southampton, Professor Karen Temple and Professor Deborah Mackay. and they study imprinting disorders, so these rare diseases that lead to serious phenotypes. So these epigenetic signals are laid down early, but if there's any abnormality in these, as I mentioned, they lead to serious phenotype. They've been trying to unpick what is causing these epigenetic failures, and what they've found is, in fact, there are some genetic causes that lead to these abnormalities of imprinting. They identified that in mothers, there's mutations in this gene here, NLRP5, that can lead to imprinting abnormalities, as well as in children, this gene here, the zinc finger protein 57. And so all these discoveries made in Southampton. They've identified in children that they can see multiple imprinting effects, that they're really likely to show a mutation in this gene. They look at the clinical effects, obviously significant changes in growth, development, metabolism, behavior. And they've been able to define new syndromes, such as Temple syndrome. And obviously all of this information aids in treating these patients better. Also, as you can see, they've published a huge number of publications, very high-level journals, so highly successful in this endeavour. So this is just a collection of these imprinting disorders. And as you can see, there's a large number of these. But what's really interesting about these is not only is the rare disease itself, so we've got transient neonatal diabetes, Seville-Russell, Temple syndrome, Prader-Willi, Angelman, Wang-Kagami, Beckworth-Wiedemann, pseudohyperparathyroidism B1. But what's also interesting is these individuals are actually at risk of other common diseases. So there may be some interesting biology that we can learn by studying these rare disorders. They obviously accurately phenotype all these individuals, and we can use all this information to essentially, hopefully, unpick some fascinating examples of human biology. Thank you.
What we also know is that these methylation marks are set up on our genes in early life. So we know that upon fertilization, the methylation marks, first on the DNA of the dad in blue, then on the DNA of the mum, are largely erased. Then as development, differentiation proceeds, there is a gradual increase in methylation, and genes that are not required in a particular cell type are methylated and switched off. And this creates tissue-specific patterns of gene methylation and expression. And it is this which essentially creates our distinct cell types. But once this period of development plasticity is over, it is thought then these methylation marks are relatively stable and maintained throughout our life. But about 12 years ago, when the IDS building was being designed and built, we had various conversations in Southampton about how stable were these marks. Could the environment affect these marks? Because we thought this might be a mechanism to explain how the environment can actually affect disease risk. And we thought, that this because we knew that the supply of methyl groups was largely dependent upon dietary methyl donors and cofactors. So we thought that variations then in maternal diet or nutrient availability in early life might perturb the establishment of these marks, might lead to a resetting of these marks on our DNA, leading to long-term changes in expression of genes and alterations in the metabolism, leading to an altered disease risk. So we wanted to test this, whether variations in maternal diet could alter the methylation of key metabolic genes within the offspring. So we fed pregnant rats a modest reduction in protein during pregnancy, and we found this altered the methylation of the PPAR alpha gene. So what we found, there was a reduction in methylation across the promoter region of this gene in the liver of the protein-restricted offspring compared to the control offspring. This decrease in methylation of PPAR alpha was accompanied by an increase in the expression of this gene and an increase in levels of beta-oxidation, the metabolic process that it controls. We found this change in methylation occurred as early as embryonic day 18 and persisted through the life course of the offspring. But we also found that it wasn't just maternal undernutrition that could induce these epigenetic and phenotypic changes, it was also maternal overnutrition. So here we fed pregnant rats a diet either high in saturated fats or in fish oil, and we found this altered fatty acid composition within the liver of the adult offspring. We saw a decrease in levels of 22, 6n minus 3, and 20, 4n minus 6 levels in the liver of the offspring from those dams, fed the diet either high in saturated fats or in fish oil. Now, the FADS2 gene is the gene that is primarily responsible for the synthesis of these fatty acids. So we then looked at its expression and methylation in the offspring. And what we found was there was also a decrease in the expression of FADS2 in those offspring from the dams fed the diet either high in saturated fats or in fish oil. And this decrease in the expression of FADS2 was accompanied by an increase in the methylation of CpGs within the promoter of this gene. And we found that methylation at CpG 394 accounted for over 56% of the variation in levels of FADS2 mRNA levels in those individual offspring. So what then about humans? Does the same phenomena occur in us? So with colleagues at the University of Edinburgh, we looked in the Motherwell cohort, and we looked to see if here maternal nutrition could alter the methylation of DNA within the offspring. And we found higher maternal protein intake or vegetable intake led to the methylation of the glucocorticoid gene. And interestingly, we made these measurements in adults who were 40 years old, suggesting again that variations in maternal diet could induce these very persistent epigenetic changes within the offspring. Subsequent studies have shown that it's not only the diet of the mum that is important, but also the diet of the dad. So if you take a newborn infant from either a mum or a dad who have had a high-fat diet, what you find is widespread changes in the methylation of their DNA. This change in methylation is accompanied by an increase in fat mass of the child, which then tracks into childhood, adolescence, and back into adulthood. And this creates, if you like, a cycle of obesity. So what we know then is diet in early life is very important, and it sets up the methylation of the DNA of the child. But we're also beginning to learn about other exposures that can also alter the methylation of the offspring. So Keith Godfrey has shown that high maternal BMI, excessive pregnancy weight gain, parity, age, can all again alter the methylation within the offspring. And what they found here was these are risk factors which predispose the child to an increase in adiposity. And what they found that 2/3 of the CpGs that are associated with an increase in adiposity are also associated with an increase in these risk factors too, suggesting that these factors influence the adiposity of the child through altering the epigenetic regulation of genes. So we're beginning to learn then from both animal and human work that early life in environment can alter the methylation of key metabolic genes. We know that changes in methylation of these genes appear to persist through the life course, contributing to changes in expression of genes, metabolism, and disease risk. So if this is the case, then it should be possible to detect such methylation changes in early life and use these then to predict the metabolic capacity and the disease risk of the offspring. The only potential issue with this in humans is there is limited tissue availability. So readily available in tissues would include cord blood, placenta, buccal, or blood. And so there's a real question mark whether the methylation in these cell types adequately reflects the methylation in more metabolically relevant cells. But maybe if the environmental constraint occurred early enough, then all three germ layers may be altered. So we decided to look to see if we could find methylation changes at birth associated or predictive of later adiposity. So back in 2011, we initially used a candidate gene approach. So we sequenced the promoter regions of genes known to play a role in adipogenesis in cord tissue from 2 mother offspring cohorts. And we found that the methylation of the CpG within the promoter of the retinoid X receptor at birth was associated with an increase in fat mass of children aged 6 in the Southampton Women's Survey and age 9 in the Princess Anne cohort, suggesting then it may be possible to detect these changes in early life and use these to predict later phenotype. As such marks might be extremely useful to identify individuals at increased risk of disease, we then carried out a genome-wide screen. So we looked for methylation differences across the genome associated or predictive of percent fat mass of these children aged 6. We used here a methyl capture approach, and from this we found 93 differentially methylated regions at birth that were associated with percent fat mass of these children aged 6. We found that the top pathway enriched amongst these DMRs was DNA replication and repair, with one of the DMRs being found within the CDKN2A gene. Now, this is a gene which encodes for 2 cell cycle inhibitors and a long non-coding RNA called ANRIL. So in order to validate this association, we then sequenced the CpGs within the DMR in an independent group of children. And we found, consistent with the array, that lower methylation of CDK at birth was associated with an increase in fat mass of these children aged 6. We also saw a similar association at 4 years, but no association at birth, suggesting that maybe altered methylation of this mark may be involved in the gain of fat mass. Now, 2 pathways to obesity have been suggested, one path being linked to impaired fetal growth and followed by rapid catch-up growth, the second pathway being linked to fetal overnutrition and an increased fat mass at birth. So we wondered then whether the methylation of CDK might be associated with this first pathway. So we looked at the association between CDK and fetal growth, and we found that lower methylation of CDK at birth was associated with impaired fetal growth from week 34 of gestation to birth, suggesting that maybe it is a mark of this first pathway. We have since replicated this mark in 3 independent cohorts, in 3 different tissue types and across time, suggesting that maybe this mark then is a robust marker of adiposity. And Keith is going to continue.
So Karen's shown you some of our data for childhood adiposity, and that's a really important focus of our work. The effect sizes are really quite substantial. We see 25% of the variance in childhood obesity explained by single marks, 2.5% explained by a bundle of fixed genetic marks, so powerful effects. But we need to look across a range of phenotypes, and that's what we've been doing. Cyrus, perhaps this morning, might have argued that we were being a bit lacking in focus. But actually, the reality is if we're going to do interventions, which we're now doing, we want to improve adiposity, we want to protect brain development, we want to protect against asthma development. So we have been looking across a range of phenotypes, and we find that sometimes similar and sometimes different epigenetic biomarkers are associated with those phenotypes, and always going on to replicate in independent cohorts. Now, for some of these marks that we're seeing, there are clear, straightforward prime drivers, but for others, there's much complexity into what's underlying the methylation changes. So this is a slide from John Holloway, where as part of a major epigenetic consortium, PACE, Pregnancy and Childhood Epigenetics, the effects of maternal smoking on the offspring epigenome have been assessed. And undoubtedly there are a large number of changes associated with maternal smoking, many of which persist beyond the neonatal period into later life. We've also been going to other cohorts, like in Singapore, where I've just come from this morning, where we have 3 different ethnic groups that we study. Chinese, Malay, Indians, and if you try and separate those using principal components analysis of their genetic data, it's fantastic. We get a perfect separation of those according to genotype. If you do the same by epigenotype, it's a starry sky. There's no real clear separation across these statistical techniques. We've gone on to look at the interaction of genetic variants and environmental exposures in relation to these kids that we've been studying. So certainly we can find bits of the epigenome which are clearly influenced by fixed genetic variants. And these 1,400 variably methylated regions that we identified, we found that around a quarter were explained by genotype alone, whereas 3/4 of them were explained by the interaction of genotype with perinatal environmental influences. And akin to that, some more of John's data shows that there are indeed these complex interactions between genotype, smoking, in relation to lung function. So for some genotypes, you don't see any effect of change in methylation on the outcome. This is a lung function outcome. Whereas for other genotypes, you see that the methylation level is very strongly associated with the phenotypic outcome. So in putting this all together, we used to have a simple sequence from genome through epigenome, transcriptome, proteome, down to phenotype. But the real picture is much more complex, where the genome is affecting the epigenome, the expososome, the metabolome, is affecting the epigenome, and particularly now exposures relating to our microbiota are being defined as having important influences on hepatic epigenetic processes, for example. So it's this complex landscape in which we've got to try and move things forwards. And we've been studying the epigenome in the Singapore infants serially from birth using stool samples, looking at the microbiota in its totality. We find that there is a number of influences that relate to the rate of acquisition of a more anaerobic adult-like microbiota. And we're starting to define these different patterns which normal human infants and others that have had C-section delivery, being born premature, treated with antibiotics, they have different patterns of microbiota acquisition, some of which seem to have lasting consequences for adiposity for certain and probably for other outcomes. So another question that I think was in this morning's session was how, what's the point of all this research? Where are we going with it? And I think part of the point is intervention, be it public health interventions, be it pharmaceuticals, etc. And of course we can intervene later in life in vulnerable groups, but the push is to fundamentally change the trajectory through intervening earlier, and particularly now looking into childhood and adolescence, that group being the parents of the next generation. So what are the exposures that we might intervene in relation to? So using our observational cohorts, we've been defining a bundle of exposures on one side in Southampton and another side in Singapore that relate to childhood overweight and adiposity. And these risks that we see when multiple risk factors are present, 4 or 5 in Southampton, 11-fold odds ratios in Singapore, where we have gestational glucose levels. These are pretty substantial risk factors for childhood overweight and obesity. And we're now moving into interventions using epigenetic marks as one of our outcomes to see if we can change things. So one of the big drivers, we think, is the glucose levels of the mother during pregnancy. High glucose levels just flood across the placenta. They get laid down in fat in the babes. They're fat at birth, and sadly they're fat throughout childhood. And a range of metabolic processes are altered, the so-called developmental overnutrition hypothesis. One foray into this is a randomized controlled trial we did with Lucilla Poston in London in 1,500 obese pregnant women, where a health trainer linked with the mothers, 6 visits, reduced the glycemic index of the diet during pregnancy. And although we didn't change gestational diabetes, the offspring were thinner by around 1/4 of a standard deviation at age 6 months. And one of the interesting facets of that is in the top here. The intervention effect was confined to those mothers who breastfed for more than 3 months. And it may be that this is a prenatal intervention which is having its effects through changes in the nutritional or even the epigenetic milieu that is there in breast milk and goes across to the baby. So another layer of complexity. In Southampton, over the last few years, one of our big areas of activity has been a new preconception randomized controlled trial, UK, Singapore, New Zealand, and where we've been intervening with a nutrient drink that has 3 groups of ingredients. Myo-inositol is a natural thing that we all make, which increases insulin sensitivity. Probiotics have been linked with a reduction in gestational diabetes, and we've identified a number of micronutrients which around half or 2/3 of Southampton women are deficient in, and which are associated with higher fatness of the child 6 years later. So this nutritional drink we've been trialling twice a day, preconception in pregnancy, very detailed phenotyping before and during and after pregnancy. It's been a huge activity to do it. We've now recruited 1,700 or so women pre-conception. We've achieved our target of more than 600 now pregnancy. Primary outcome to maintain healthy glucose metabolism during pregnancy, with the aim of lessening the risk of childhood obesity. It's a straight RCT with a yes/no outcome for the primary outcome and for secondary outcomes. But we've collected over 800,000 biosample aliquots of stool, placenta, blood, urine, etc. So it's a big discovery platform for the next generation of studies. So to wrap up, have we achieved anything in epigenomics over the last 10 years in the IDS? I think we've really moved the field forwards. We've been leading the world in some aspects of that. The animal studies show for certain that nutrition can alter the epigenetic processes. We still have a big task to define the driving exposures in humans, but we've got pretty good insights into those now. We think that the epigenetic changes do provide at least part of the memory of development, but we actually need to gird our loins and not just do one NIPA type trial, but we need a whole raft of new research trials. If this was cardiovascular disease, there would be hundreds of pharmaceutical interventions. Here's something with much more powerful, lasting effects, and we've only got a handful of trials worldwide. So what we need is people who are fired up to commit their lives to taking forwards this important agenda. Thanks.
Thank you very much indeed, Karen, Chris and Keith. I think we've got time for one or two questions. Let's see how it goes, because you probably forget them, don't you, over the course of an hour or two. Who would like to quiz this group on epigenomics? I've got one question for you, which is that, so you're going to come up with these wonderful predictive markers, and you're going to understand the processes, perhaps in early life, which have tweaked these epigenetic changes and set up risk. But are they reversible, these changes? You know, even in old geezers of my age, can you do something about it, Doctor?
So, do I need that? So we're directly doing that at the moment. So one of the things we've done is done muscle biopsies from the Hertfordshire cohort study, 60, 70-year-old adults. We've got out, Karen and the team have got out the primary myoblasts. We found that there is an epigenetic signature of low muscle mass, low muscle function. We found that it persists as you take those myoblasts out through the cultures, and we're exposing those cultures to pharmaceutical and to nutraceutical agents to see if we can alter the epigenetic signature. And we're already starting to see interesting changes in the function of those cells. So a lot of epigenetics is reversible. Your epigenetics now are different to when you woke up this morning. And in fact, your diurnal cycle, your clock genes, etc., they're just cycling over a period of hours. So some marks are changing a lot, whereas others are probably changing very little from the time we're conceived to the time we die. So there's all patterns out there.
Any other questions? Yes, Philip. Behind you.
Thanks. Karen touched on this, I think, which is the question of the cellular and tissue specificity of epigenetic marks. And I think you speculated a bit, but what is now known about how similar epigenetics might be across different tissues?
Okay, so lots of groups have compared methylation in different tissue types, and using the 450K array, which looks at only a subset of CpGs, what they've found is that basically there are sort of about 4,000 CpGs that are conserved in blood and liver, and about 5,000 in blood and muscle. So we're beginning to sort of build up a picture of the CpGs that are conserved from blood and tissue type, and then we can focus on those and look at how they change in blood, and use those perhaps as markers. But it's still a relatively young field, and we're still sort of learning. Yeah.
Karen Temple.
Obviously, we've been concentrating on methylation of DNA. Do you think that there are other epigenetic marks that are going to be more relevant, more versatile, will be better ways of really identifying the regions that could be altered by treatment in some way? So it might be that methylation is too far down the line.
Yeah, so in animal work, what we've seen is that you often get a change in the histones first, and then that is consolidated by a change in methylation. So I think that the methylation, you're right, is probably secondary in some respects, but in human it's quite hard to look at histone changes, and we haven't quite got there yet. I mean, there may be some exceptions to that, because we know that methylation in some cases can change very quickly. Exercise sort of changes the level of methylation within a couple of hours. And so, you know, and there are some transcription factors which can change the methylation of DNA as well. So, but in the animal work, I think the histones would be, you know, good place to start. And we would dearly like to look at histone changes in humans.
But we're already quite a way down the line with some of the non-coding RNAs. And one of the methylation sites that we've been interested in encodes for a long non-coding RNA. We saw data just yesterday in Singapore from one of the studies linking a circular RNA to some of the changes we're seeing in muscle. And we're looking at the moment at some of the small transfer RNAs in relation to sperm. So it's a fantastic Pandora's box that has been opened up, and what we need to do is use it appropriately to actually progress new pharmaceuticals and new public health interventions. Yeah.
Thank you very much. So I'm not sure whether to be comforted by that or not. My tree of life over here is looking a bit sad, and if you're not telling me that you'll be able to run this epigenetic clock backwards fast enough to help me, I'm going to ask for another strategy, which is to maybe think about starting at the bottom again. Could we actually, by reprogramming stem cells, actually replace some of the dodgy bits of my body, of which there are many, and therefore give me a healthier tree, which of course is the introduction to our regenerative medicine group led by Professor Richard Arefo. And because they're a young developing stem cell group, I gather that it's actually the young developing stem cell students that are going to give the presentations. Is that right, Richard? Or are you going to introduce them? Good, thank you very much.
Good afternoon. My name's Richard Arefo, and here at the Centre for Human Development, Stem Cells and Regeneration, we're focused on trying to understand human development, tissue regeneration, and what stem cells contribute to that process. So we've heard throughout today the importance of epigenomics, genomics, developmental physiology and nutrition, and indeed, as you can see here, this, I hope someone's getting royalties for this slide. You can see here this wonderful increase in life expectancy. We can all hope to live to our 8th, 9th, and 10th decades. But of course, as Mark has alluded to, that provides challenges in terms of, I think, withering tissues was what you were talking about. And just think about this. Within the next 2 years, 20% of the population will be over 65. So the vision in the centre is to actually translate pioneering developmental science and stem cell science for patient benefit. It's this ability to try and address some of those issues with longevity that will occur, that reduced quality of life and changes that are associated with aging. And we're going to use the examples of bone and cartilage and how we can use stem cells to actually regenerate those tissues. That's the examples we're going to be using within the centre. But the centre works across the whole spectrum of tissues. And in fact, let's just remind ourselves, this just shows you this dramatic increase in aging. So it's only in the last 50 to 70 years that we've actually seen this dramatic increase in longevity. And stem cells, we hope, will transform patient lives. And the reason is because stem cells have this unique capacity. They can generate a whole range of tissues. I've talked about bone and cartilage, near and dear to us, but also, of course, we could be looking at liver, or we might be looking at cardiac, or we might be looking at nerve cells. Within the centre, we've pioneered the collection of fetal tissue. We've actually worked on human embryonic stem cells that helps us to understand abnormal and normal development. And what we're going to show you is a series of vignettes presented by the postgraduate students within the centre, and then we'll close with a clinician that's actually going to tell you how we've managed to move right through to the clinical spectrum. And so I'd like to start with Lauren Javance, who is a PhD student who's studying human embryonic stem cells and cartilage regeneration. Lauren, please.
Thank you, Richard. So I'm going to introduce you to stem cells and discuss how we can harness their potential for regenerative medicine. So stem cells are an unspecialized cell type with the ability to self-renew. And they can differentiate to become progressively more specialized tissues. These stem cells form a hierarchy. We start with the totipotent cells that can form all the embryonic and extraembryonic tissues. And then we restrict the potency and we have pluripotent stem cells. These can become any cell type in the human body. They can be derived from the inner cell mass of blastocysts, or they can be generated from patients by reprogramming somatic cells. As we progress down this hierarchy, we have multipotent adult stem cells whose lineages are restricted to just a few cell types, and then unipotent cells that are progenitor cells that can be just one cell type. Now these cells are amazing because they have the potential to repair and regenerate damaged tissues. Now for my work, I'm interested in pluripotent embryonic stem cells. These cells are truly amazing. They are derived from the inner cell mass of blastocyst stage embryos, as you can see here. And as they're pluripotent, it means that they can generate any of the somatic cells that are in our human bodies. And they proliferate indefinitely in vitro without showing signs of cellular senescence. And this is an issue associated with adult cells. In culture, they grow in these nice, tight, round, compact colonies, and they express the key pluripotency markers, OCT4, SOX2, and NANOG. Now these characteristics, such as pluripotency, the fact that they are not patient-specific, and that we can yield very high numbers when growing them in the lab, and they proliferate without showing cellular senescence, make them an ideal cell source for regenerative medicine. Now I want to use these cells to treat one of the biggest problems facing our joints, osteoarthritis. This is a disease that is the most common joint disease, and it's an example of withering tissues, as we've heard about today. It's caused by cartilage degradation, and it's a debilitating, life-altering illness that causes severe pain to the individual, and it dramatically reduces mobility. As osteoarthritis progresses, small cartilage lesions, they worsen. You get increased cartilage loss, and ultimately patients require total joint replacement surgery, which is not the ideal situation, because what we need is an effective early intervention treatment. But currently these are lacking. There are some pathways available, such as transplantation of the patient's own cartilage cells into the affected area, or drilling into the cartilage to try and encourage cells from the underlying bone to repair the damage. But these treatments are inadequate because they typically produce a very rough, fibrous cartilage rather than the smooth form that should be covering the ends of our bones. And patients typically undergo various iterations of these treatments before ultimately requiring total joint replacements. So what we want is an effective early intervention treatment. And I think that embryonic stem cells may be the answer. So we have shown that we can differentiate human embryonic stem cells into cartilage cells. We've worked on a protocol that takes 14 days. It's based on the developmental pathway of chondrogenesis. And we supplement the cells with a variety of growth factor cocktails, and we start with our pluripotent cells at day 1, And by day 14, we have cartilage cells. Over the course of this, we see there's a decrease in the markers associated with pluripotency and an increase in markers associated with cartilage, such as SOX9 and type 2 collagen. And amazingly, we find that these cells are actually capable of forming cartilage in vitro, which really highlights their potential as a future early intervention treatment for osteoarthritis.
Thank you, Laura. That's just giving you an example of human embryonic stem cells. Wow. Okay, so we're also interested in the center in adult-derived specific stem cells. And Miguel Xavier is just completing his PhD. He's been using a variety of interdisciplinary approaches, and actually he's been using microfluidics. And it, in essence, demonstrates the collaboration across the faculties between the center and, in particular, the Institute for Life Sciences. So, Miguel.
Thank you very much, Richard. So, indeed, adult stem cells have limited capacity when compared with the embryonic stem cells that Lauren has just introduced us to. But they have also a very big advantage, which is the fact they can be sourced from the own patient supply of stem cells. And this will avoid any issue with tissue rejection. Specifically, I'm going to talk about skeletal stem cells, as these cells have the capacity to form fat, bone, and cartilage, and they can also be grown in the lab and used in tissue engineering approaches to form new bone and cartilage that can be implanted back into the patients. However, there is one big disadvantage with these cells, which is the fact they are extremely rare. So it is estimated that fewer than 1 in every 10,000 cells in the bone marrow is actually a skeletal stem cell. And the current methods for their isolation do not provide the purity required for their routine clinical translation. So at the IDS, we are looking into microfluidics as an alternative approach to sort these cells from bone marrow. Microfluidics are able to sort these cells based on their biophysical properties. But to do so, we first need to find which are the biophysical properties of skeletal stem cells that allow us to actually discriminate them from other cells in the bone marrow. For that, I'm going to take you to the trip to the supermarket. So when you go to a supermarket, take these two kiwis. How would you choose the right kiwi to take home? When you look at them, they look exactly identical, but actually the answer becomes quite clear. You apply a mechanical test, and it's obvious which one is good to take. And that is what we are doing with the skeletal stem cells. So using a technique called real-time deformability cytometry. In this video, what you see is flow coming to the right to the left. And you can see that cells, when they go through that narrow constriction, we are squeezing them basically one by one. And you can see that the cells go from this spherical shape when they are in suspension into this kind of bullet-like shape when they are deformed. Using a high-speed camera, we can quantify how much these cells are deforming from this perfect circle. And then we can plot data as the one that you see in the graph. And more interestingly, we can actually compare different kinds of cells. So we have mixed skeletal stem cells with white blood cells, and we have shown that the skeletal stem cells are stiffer than white blood cells, and white blood cells are very abundant in the bone marrow. So this was good, because this is something that we can use to sort the cells. We were not sure if it was enough, and actually most of the microfluidic techniques are based on cell size. So we wanted also to have a very accurate measure of the size of skeletal stem cells in bone marrow. And for that, we use another technique. It's called microfluidic impedance cytometry. It uses non-harmful electric fields. As cells flow through this electric field, they are going to perturb the electric field, and you can measure that. And basically, we can plot graphs of data that look like that. And we have cell size in the x-axis, and each dot in these graphs represents one single cell. The skeletal stem cells are here represented as the red dots, and we can actually see that they are contained within the larger fraction of cells within the bone marrow. So that's good. So we know these cells are some of the largest cells in the bone marrow. We also know they are stiffer, so how can we use this to actually sort cells? And that's what we're looking into right now. So using now a microfluidic sorting device, this device is able to sort cells based on size, and you can see on the first video 2 different cell populations being separated because they are different in their size, and also to sort cells based on stiffness. So these are red blood cells flowing in one of these devices, and red blood cells are quite soft, so they deform quite easily. And you can see when they interact with the pillars how they squeeze around these pillars. So it affects and changes their apparent size, allowing for separation. And we can use these devices to sort cells at throughputs of 1,000 per second, which is very important when you're trying to sort rare populations of cells, such as skeletal stem cells in bone marrow. So our goal is to have samples from patients, which we know are very heterogeneous, but just by running them through one of these devices to obtain pure populations of cells, which can then grow in the lab and using tissue engineering approaches to grow new bone and new cartilage to be implanted back in patients and hopefully improve people's lives. Thank you very much. Richard.
Thanks, Miguel. So, once you've actually isolated your cell, it's actually pretty important to understand the regulatory and signaling networks that control that cell. If you have a cell that has that potential to form any tissue type, it's very important that that cell only makes the tissue you desire at the site that you desire. So an understanding of the regulatory network, of the signaling pathway, is pivotal if we're then going to apply those cells in a reparative paradigm. And Patrick Stumpf has just completed his PhD. He's an early career researcher, and he's developing novel modeling approaches to understand the gene and the signaling networks in stem cells and how we can characterize these cells and then how we can start to manipulate and apply those characteristics to those cells. Patrick.
Thank you, Richard. So thanks to recent technological advances, we have entered a new era of quantitative single-cell biology. And using a combination of droplet microfluidics, up here, and next-generation sequencing, we can dissect complex tissues such as the bone marrow into its building blocks, cells. And we can resolve cell identities based on the gene expression signatures alone. This data enables us to examine the regulatory circuitry within individual cells, and we can look at essentially how the logic in these regulatory networks is altered given a cell's identity. So here shown 4 different cell identities. And we can also ask and possibly answer how the circuitry is altered by aging and by disease. Using similar single-cell methods, we can study how individual stem cells adopt distinct cell identities. So going from the stem cell to the somatic cell. And from this data, from the single-cell data, we can reconstruct collective cell behavior in time. And our objective is to model this dynamic process using mathematical methods. And therefore we apply theoretical models that we adopt from statistical physics, and they help us understand how stem cells adopt different cell identities and also how they age. And that's everything for me.
Thank you, Patrick. So, as well as understanding, I don't know if you saw, if we just go back one slide here. I don't know how many of you have been outside and have looked at the stem cell mountain. Okay, so I'd encourage you to go to that later on today. We're actually passionate about public engagement, and in the centre and across the whole institute, and you'll hear a whole lot more about that. I'm just going to show one slide from Professor Hansen. So this is the stem cell mountain, and we use this to actually describe and to take to festivals. It's been to more than 30 festivals, been seen by more than 300,000 people. It's been to Glastonbury. It was developed with the Winchester Science Foundation. John Dawson, Nick Evans, and myself are involved in this. And essentially what it allows you to do is to imagine the stem cell as a marble, or the marble as a stem cell, and you can actually, at the top of the mountain, that marble can move in any direction. It's pluripotent. And as it cascades down, it becomes restricted, and we use this to try and explain tissue differentiation and lineage restriction. I also hope you have the opportunity to go and look at some of the augmented reality that's present outside, developed by Stuart Lanham. And again, that's useful in trying to get across to the public how the work that we're undertaking can be understood. So as well as enthusing the next generation of scientists, we have a vibrant postgraduate program here. We have a new Master's in Research in Stem Cells Development and Regenerative Medicine. It was developed by Francesca Houghton and Dr. Ralter. It started last year. We also have a 4-year integrated PhD in stem cell research. But it's not just the postgraduate community that we've tried to enthuse. We also engage with the undergraduate community. So this is a module, this is an interdisciplinary module, engineering replacement body parts. It's multi-faculty, so there are actually 3 faculties involved in this. Engineering and the environment, Alex Dickinson, I think, is also in the audience. Natasha Hammond-Browning and Tracy Newman, business and law and in medicine. And it's led by Nicholas Evans from the centre. And this module covers stem cells, law and ethics, bionics, prosthesis, and tissue engineering. Tissue engineering. The ability to take your stem cell with your scaffold of interest and growth factors and cues and create the niche, the environment that tells that cell to form the tissue of interest. And it's pivotal that we can try and develop those niches because that's what leads to translation through to the clinic. And I'd like to introduce Mohammed Musa, who's a first-year student who's working with nanoclays to generate the appropriate environments for our stem cells, to develop our stem cells, and to actually generate tissues of interest. Mohammed.
Thanks, Richard. Thanks, Richard. So, hi everyone. So, following what my colleagues have explained to you about the stem cells, their potential, their types, and the methods and developments we have for their isolation and culture expansion in vitro, comes a very important question, which is, how can we control the stem cell microenvironment or niche? And how can we safely transfer or translate our stem cell-based strategies from the lab to the clinic? To be honest, and in fact, during this process from the lab to the clinic, we face many challenges, the key of which is number 1, how can we deliver, as you can see on the left, how can we deliver a stem cell with the inductive signals, and how can we hold them in the right targeted location into the body? So this is the first challenge. The second challenge is how can we localize and hold these biological signals, which are important to induce the stem cells to differentiate into the desired tissue. How can we localize them in the targeted location into the body? Because, you know, when these signaling molecules are injected, they start to diffuse away, and in turn result in severe side effects like bone formation in the wrong place, swelling, and osteolysis. And here comes the role of clay. They say that man was created from clay, right? Anyway, so what we have found is that our nanoclay particles, under physiological conditions, they have the ability to self-organize into something called a thixotropic gel. This clay gel has the ability to encapsulate cells. As you can see here, for example, this is single cell encapsulation within our clay microcapsule, and these are viable cells in our clay gel. Also, and uniquely, due to their thixotropic properties, which means that our clay, the viscosity of our clay is responsive to the force which is applied to it. This means that they can be easily injected into the body, and after injection, post-injection, the clay nanoparticles reorient themselves to re-establish a gel which is sufficiently cohesive to bridge the gap between a bone fragments, which is really important for something like, for example, a bone fracture repair. And this is a video to show you our clay gel and how can we prepare it. For example, this is the passage of our thixotropic gel through a low gauge needle in PPS. And not only this, when it comes to the second challenge here, which is, for example, when we look at a growth factor called a bone morphogenic protein, this is widely applied in orthopedic surgeries. But the problem with that protein is that high doses are required for efficacy because according to what's used now for the biomaterials that is used to delivering it, they cannot localize this protein in the right targeted location. They start to diffuse, which means that we need high doses of these growth factors and in turn severe side effects. What we found is that our clay gel can induce bone formation at super low doses of these growth factors, which is around 3,000 lower than what's currently employed in clinical practice. As you can see, for example, here, the osteogenic differentiation of the cell is localized only to the clay compared to its clay-free control, and here in vivo, compared to the alginate, which could not support bone formation at high doses of growth factors, at low doses of growth factors, our clay sustained ectopic bone formation at both the high and 40 nanogram super low doses of growth factors. And more interestingly, based on these exciting data that were presented to you, we have a new Southampton spin-out, Renovus, for which we have an Innovate UK grant to transfer our unique nanoclay-based strategies from the lab to the clinic with lower cost and minimal side effects. Thank you.
I hope Mohammed has convinced you of the importance of the environment. Okay, so not just creating the right environment for the cells, it's vital we have the right environment for our students and as a community. And this beautiful slide from David Wilson just shows you the international reach that is present within the centre. We have collaborations across the globe. We have a joint stem cell lab with the Chinese University of Hong Kong. And we are a diverse and vibrant research community. We have more than 40 PhD students from the Americas, Africa, Europe, and Australasia. We are international. Okay, so I'm just going to close by just saying at the start we mentioned this desire to live to our 8th, 9th, and 10th decades, but of course, that brings its own challenges. And this is, for example, one of the biggest challenges we face in the musculoskeletal field. Just think about this. Every 3 seconds, a bone fracture occurs worldwide. Okay? What we need are simple strategies. This is back to Mark's point of how we're going to stop the withering and how we're going to stop any of these detractions in terms of our physical life. Simple strategies for bone and cartilage repair, whether we're talking about spine, tumour, trauma, fracture from osteoporosis or osteoarthritis. We know that 1 in 3 women, 1 in 5 men over 50. Now in the UK, just have a look around the room, okay? Have a look around the room. In the UK, 1 in 2 women, look around, 1 in 5 men, you feeling lucky? Okay, will suffer an osteoporotic fracture after the age of 50. Okay, so the numbers involved and the socio-economic consequences are truly devastating. What we want to close with is actually not one of our students, but actually a consultant orthopedic surgeon that's been working with the Bone and Joint Research Group for more than a decade now, Mr. Douglas Donlop, who has actually translated some of the basic science that's been undertaken within the group in terms of patient benefit. Okay, Douglas.
Thank you very much. It's been a great pleasure working with Richard over the last 12 years now, I think. Because not only am I working in a teaching hospital, which is fantastic from a teaching point of view, but being able to translate the basic science for patient benefit has been brilliant. Some of these pictures up here just show the breadth and depth of orthopedics. It's just fantastic. You can instrument somebody's spine, you can treat cysts in children or cancers in adults, or you can look after people who've been unfortunate enough to arrive in the hospital in more than one ambulance. That's usually a bad sign. And fragility fractures, which is what he's been talking about as well. When I started 15 years ago, there were 16 of us. I was the 16th. There are now 32 of us. And so the increasing ageing population is definitely having an impact on the orthopaedic department. We've already sorted out the problem that can happen to the femoral head here. It can die if you have a fracture or a damage to the blood supply, and we've translated using stem cells to regenerate the femoral head. So we've done that. We've also looked at using cells to improve bone graft, which we use for revision hip replacements. So it's happening, and it's very exciting from my point of view. Just to touch on the mechanism, here you have somebody who comes in with a broken leg. If they're young, you could treat this just with traction, because they have the biological potential and the patience to heal on their own. Most adults want to get up and walk about, and if you're elderly, then you may not have the biological potential to heal the fracture. A broken bone needs to have stability and biology for it to heal. And we now know that you can make these ununited fractures heal by putting stem cells into the fracture zone. We know that they have to have a concentrated number of a certain type, and it's this that's been particularly exciting to become involved in. Trying to identify which of the patients who are going to develop this with tests and markers and things that you can put into the area will obviously be exciting too. Of particular interest to myself, I'm a hip surgeon, and when John Charnley invented the hip replacement in '66, you usually had to be almost crippled and at least over 50, whereas now 80,000 hip replacements were done in the UK last year. There isn't an age limit, except that you should have probably stopped growing, so you usually have to be an adult. And if, in this case, this lady was in her 30s when she had her first hip replacement, she then had a number of revisions. When she came to see me, she had this massive hole in the side of the pelvis, looked like she'd been shot with a shotgun, and we originally treated it with some bone graft using the cellular augmentation which we developed here with Richard Arefo, and that did well from 2004, I think, 2002, sorry, until 2014, but then it failed. And then we had an even bigger hole, and what were we going to do with that? So bone grafting is not stable enough. You could put biology in it, but it wouldn't be stable enough for long enough to recover. So we were designing large three-dimensional hip replacements. I've now done 15 of the things, more than anybody else in the UK, and the key with this, which is different to other implants, is that we wanted it to bond to the patient rapidly, because they're often elderly, and the implant is massive, and the bed that you're putting it into is often sclerotic and doesn't have a good blood supply. So we started coating these with stem cells, which we harvested from the patient beforehand. We got some notoriety, got onto Sky News, and it's particularly relevant in these older patients because this is a CT scan looking at their bone density profile, and all the green stuff is rubbishy bone, which you can't really get a screw into. And so even though you have 16 screws in this, you do need something extra, which is what Richard has managed to provide in my particular situation.
Thank you, Deb. So I'm just going to close by saying that indeed we think the future is bright for stem cells development and regeneration. We're starting to unravel the key regulatory mechanisms important in these stem cells to isolate and enrich for human embryonic, for induced pluripotent stem cells, for adult stem cells. We're starting to generate the niches pivotal in translating from preclinical through to the clinic. And we've only given you one story focusing on the musculoskeletal from the center. The center works across the whole spectrum, from cardiac right through to nerve, right through to nutrition in terms of the early embryo development. And this is just to give you a flavor of that. It's really important to acknowledge everyone within the center. I realize, as I say, we've only focused on one piece just to get across the story and how we can intervene across across the life course. But actually the most important thing, and I hope you've seen this with the postgraduate students, that none of this would be possible without the research fellows and the postgraduate students who make it possible. And finally, to thank the collaborators, both in the faculty, across the faculty, Institute of Life Sciences, and across the world, that allow us at the centre to do what we do, and within the Institute of Developmental Sciences led by Mark and Karen. And finally, the collaborative work is funded through the research councils, Wellcome Trust, a raft of charities, and of course, the European Union. And thank you for your time.
Thank you very much indeed, Richard, and the whole group. Wonderful enthusiasm there. I feel much more cheerful now. Some questions. We have 5 to 10 minutes, I think. Looks like you've stunned them, Richard. Don't you worry that you're just going to produce cancer under some circumstances? I mean, you start growing cells, that doesn't sound too good to me.
So that's a terrific question. It's really important that we have the right models that allow us, because as Mark has just alluded to, you have a stem cell that can form any tissue type. So one of the things that's really important is that we have the right protocols that allow us to form the desired tissue of interest. And yes, you don't want a teratoma, you don't want a different tissue type, you do want to have your cell forming the tissue of interest, indeed.
So,
Cyrus?
yes, so
can I bring the colleagues up, because it's your show as much as anyone else's.
I guess they were all wonderful presentations. Well done to all of you. I suppose to go back to cartilage, which was, say, 12, 15 years ago, the one that was going to break through in terms of autologous and then, regenerative chondrocyte transplantation. That sort of Swedish trial and then the Oswestry study were a little bit, a little bit sort of limited in the extent to which they suggested this was going to become a routinely available competitor in early neo-A or early cartilage defects to compete with, say, ultimately arthroplasty. So what in the developments has changed to make the step change that this might become a real sort of option? And who's it going to apply in? Because my understanding was that it might be the earliest stages of the disorder. And if that was the case, how do we identify who those people are?
So that's a really timely question, because NICE, National Institute of Clinical Excellence, have just approved autologous chondrocyte implantation, as you've mentioned, on the 6th of October. So this now means that it's a reimbursed procedure within the NHS, and it's very much due to the MRC trial that was completed at Oswestry that led to that change of mind from NICE. There is still this issue of its fibrocartilage that is predominantly present, but the quality of life and improvement scores from the patients was sufficient to swing NICE's judgment on this. I think that in terms of the tissue formed, the jury is still out as to whether it really is more than fibrocartilage, but the benefit for patient pain loss has been significant. In terms of who it would be applied to, at the moment they are looking at a much younger cohort rather than the, should we say, an aged cohort at the present time. But again, I've not seen the stratified data from Matt Spritberg's lab, for example, that would really tell us whether it's the 60 plus or currently the view within NICE that it's a younger cohort that will benefit.
Because I was sort of beginning to think that, I mean, when things go wrong in the body, especially in aging, they don't go wrong just as one single process. Douglas mentioned the fact that it might not be easy to get these grafts and transports to work very well and to stay in situ very well because the blood supply is not good enough. So if you put the stem cells in, don't you have to think about promoting angiogenesis as well? I mean, certainly in early development, there's an issue about whether growing cells draw the blood supply or whether the blood supply drives growth of the cells. So arguably, you're going about this.
Absolutely. So one of the reasons that cartilage, as Cyrus was alluding to, was, if you like, was the first choice across the community, was because it was a neural, avascular, single cell type. How hard can it be? Okay, and it's proved truly challenging to generate hyaline cartilage to put into a patient, despite the best efforts of everyone. What is really important is that you actually have in your regenerative paradigm or tissue engineering paradigm this ability to provide that vasculature, because typically you're never more than 100 or 200 microns from a blood vessel, certainly within bone. So that ability to provide nutrient supply and to remove waste and to remove toxins is critical. And so being able to prime that angiogenic process is really important, and being able to control that cascading process of how that happens, certainly within a skeletal environment, becomes really important. So one of the challenges has been that we know some of the, if you like, stimulatory molecules that Mohammed talked about in driving that bone formation cascade, but actually you have to have that in the right sequence, because if you've had your fracture and then you've had a callus and then you've had that inflammatory reaction and then you've got new bone formation, you've got to be able to stage that repair process appropriately, and that is, if you like, one of the challenges, and that's where the models become very, very important.
Great. Thank you all very much, folks. So I think it falls to me now just to say a few words and give you one or two things to think about in relation to public engagement. We've heard quite a bit about that from the various teams already today. Think of a story. Might be a story that you particularly like. Might be a story that your child, perhaps from your parents, or perhaps it's a story that you're telling your own children, or in my case, my grandchildren, not that she's old enough to understand anything yet. Now just change the beginning of that story. The whole thing goes horribly wrong, doesn't it? I'm giving you this somewhat silly analogy, this somewhat silly story, because that's precisely what I heard just a few weeks ago at a side meeting of the United Nations, organised jointly by the World Health Organization, what's called the Partnership for Maternal, Newborn and Child Health, which is a group that, working with the Secretary General of the United Nations, Every Woman, Every Child initiative, put together the global strategy for women's, children's and adolescents' health, which was published in 2015 but runs from 2016 to 2030 along with the Sustainable Development Goals. We're proud of this because actually it's the first of these UN publications that actually mentions the work that we've all been doing here over the last 10 years. The fact that in early development the risk of these diseases, non-communicable diseases, is set up. But at the partnership and WHO meeting, they used precisely that analogy. Change the beginning and you change the whole story. Why were they doing that? I think because they wanted a very simple message that could be got out to the wider public, explaining why development and starting early to prevent risk is so important. The thing about the story, though, is that you need to think where it fits in. This slide, which Mary Barker and I put together for the Lancet series, was obviously addressing the issue of adolescents and young people, the parents of tomorrow, with a view to helping them to change their behaviour and to improve preconception health. And it shows that, of course, to drive that scenario from left to right, the so-called COM-B model, capability, opportunity, and motivation to change behaviour, there are a whole range of actors involved. You need, of course, policy changes, policy priorities in government. You need clinical services and other community services that make it possible for individuals to lead a healthy lifestyle. But more than anything, perhaps, you need the motivation, the social movements, perhaps, that create awareness and demand for these kinds of services. And so it seems to me that our public engagement with our science, like a lightning strike, goes to the very heart of that. The ability that we have here in Southampton, in the university, in the institute, with our partners, to drive that motivation, that commitment, that demand, that interest. Before I say something about what we've been doing, it's also important, of course, to think about who else one is telling the story to. From this slide, of course, we've got the healthcare professionals. And actually, we've done a lot of work over the last 10 years to engage a range of healthcare professional communities. Professor Nina Modi from Imperial College will be joining us later on. She's president of the Royal College of Paediatrics and Child Health and has done a great deal, actually, to engage with the kind of work that we're doing and through her own work on the early origins of childhood obesity to really make this life course story that we've heard about across the day something which the Royal College has picked up. Of course, the Royal College of Obstetricians and Gynecologists, one would expect very much to be involved, and they have been, although, to be honest, and I don't see any obstetricians in the audience, it's until very recently been the case that most obstetricians felt that their job stopped when a healthy child was born, and they didn't think so much about the responsibility that they had for the next generation. And actually, it's not just me saying this. There's a very nice short clip from Sir Michael Marmot, who, of course, was probably the person in the UK, at least, who did more to promulgate the idea of social inequalities of health, where he says he went to the Royal College 10 years ago and they didn't understand how early life was so important for the rest of life. More recently, he's been back to talk to them and they now understand the message, and I think he's done us an incredible favour there. Of course, the Royal College of Physicians is also involved, and we've been talking to them recently. We've heard a little bit during the day about the international context of the kind of work that we do. So there are at least 2, or maybe more than that, organisations internationally that can have an enormous impact in promoting our public understanding. There's FIGO, the International Federation of Obstetricians and Gynecologists, which has members in 131 countries all over the world, linking directly to those obstetricians and gynecologists, and of course through them to midwives and other neonatal nurses, who are of course the people at the coalface who are talking to the patients. And perhaps if we're thinking about, say, some of the nutritional interventions that we were hearing about earlier on, they're the people who need to get the message across. So what's the message that we're giving them? If you go to your GP, and I know anecdotally that this is true even in this city, and say, as a young lady, doctor, I think I'm pregnant, or actually I'm planning to get pregnant, what advice can you give me? Often there's a bit of a sticky silence, as if, well, don't you know exactly what you've got to do? It's a little bit more complicated than that, isn't it? And so actually there is a real need to think about what message we're getting across to our clinical colleagues. And then the International Paediatric Association, chaired by Zulfi Bhutta from Pakistan and Toronto, where he works in the Hospital for Sick Kids, again is very much engaging with this early development story and the life course from the adolescent through to the next generation. Of course, we need to engage government, which means the Department of Health. 4 years ago, Dame Sally Davis's annual report had one single comment in it that, in fact, fetal life was very important to later health. More recently, we were able to author a chapter in her annual report, which came out in 2015, which was entitled "The Health of the 51% Women." And so Keith Godfrey, Lucilla Boston and I and colleagues wrote a chapter on preconception health for that. So Department of Health is beginning to engage with this. And of course we also talk to Department for Education, especially around our educational initiatives like LifeLab, about which I'll say something in a minute. But strangely, those two departments don't talk to one another very much either. So we've got some real issues. But I think slowly but surely we're moving forward on that. And actually the university is beginning to get much more involved in this public engagement. And we actually now have a public policy unit which across the university, which is taking this kind of thing very seriously. There are a range of parliamentary committees. I've spoken to some of them. I'm sure many of you have as well. And then there is also POST, the Parliamentary Office of Science and Technology, which has the remit of looking at some initiatives and perhaps giving a slightly more balanced and objective view of what's going on. And at the moment they are focusing on childhood obesity and the government's action plan for the prevention of childhood obesity, which is pretty thin, actually, to be honest, and we're hoping that we can really get some real mileage through there. So there's an engagement exercise at that level. There are some powerful NGOs. We work closely with the World Health Organization. Of course, there's been a dramatic change there with Dr. Tedros from Ethiopia now being the Director General of World Health Organization. He's changed most of the staff there, and we perhaps need to start to re-educate them. And of course, there have been some controversial issues in relation to some of the things that he did in his early days. But they're potentially a very influential organisation, but one, if you read Richard Horton's comments in The Lancet, are underpowered and without much in the way of strategy. So I think there's a considerable amount of work to be done. There are other NGOs too, though. For the NCD Alliance is just one that I'm currently working with. This is a partnership of hundreds, I think actually it might even be 1,000 or more, other civil organisations around the world, civil society organisations that are interested in particularly non-communicable diseases, and it gives us an opportunity to work with them across the piece. The advantage of working with NGOs as opposed to individual civil society organisations is that they have a place at the World Health Assembly, and they also have a place at United Nations meetings, and next year will be a very big year for NCDs because in September of next year there'll be another UN global strategic meeting following the one in 2011. This will be the next one in 2018 on NCDs. And so there's a lot of work to be done to engage with them to get the development agenda and the life course agenda into the NCD agenda. And of course, there are a whole range of civil society organisations. CRUK, of course, very much involved in the beautiful new cancer immunology building, which for those of you from outside, have a look at least from the outside. Soon you'll be able to go into it, but that's a wonderful investment which CRUK have been very helpful in helping us to take forward. And of course, I still have the privilege of being British Heart Foundation chair here, and BHF are again very much engaged in this agenda. DOHAD, of course, Physiological Society, and I could go on and on. We all belong to different societies. I think the more we can use them, because many of them have their own public engagement exercises, to bring them on board with what we want to do, the further we'll get. And of course the private sector, which until recently has been very much a dirty word, especially perhaps in the nutrition field. Keith mentioned Epigen, the International Consortium for Epigenetics. It's one good example of how we've been able to work across Southampton, Singapore and Auckland with major with major food companies to do research that actually is truly collaborative and not just for developing product and therefore profit for those organisations. Of course, at the end of the day, it's increasing their market share that they want to do, but there's no reason why that should not be a joint exercise. And I've been involved recently in working with something called the UN Global Compact, which is an initiative set up by the previous Secretary General of the United Nations to engage with industry. It was a rather toothless organisation, to be honest, but I think Guterres, the new Secretary General, has revised it. For example, he's thrown the tobacco industry out of the compact immediately, which is good. And so we're now beginning to think about how can we actually genuinely work with the private sector on a shared mission. Because health is everyone's business. And actually that's the name of a new platform that I've been helping them to design, which will involve a development component for sure, to bring appropriate businesses together to see not just how corporate social responsibility can deliver something that sort of blue washes or white washes what they do, but actually how a shared joint agenda can be taken forward. Then locally, we really do engage in the city. We've got very good contacts, of course, with Dawn Baxendale, who's chairman of the City Council, and Debbie Chase, who is responsible for public health. And I think we're really fortunate that in our city we have a relatively stable population. We have a great commitment, thanks to work. It's projects driven from David Barker's Environmental Epidemiology Unit initially, setting up the Southampton Women's Survey and all the publicity that's attended that. But a real opportunity, I think, to show that we can actually make a real difference in this city. So I know some of you are engaged with contacts there, but I'd urge us all to take that to the next level. Life Lab has had, I think, over 6,500 students through it now. Obviously, there are too many of us to visit it today, and I think we have a class in, or they've probably gone home by now. I'll show you a brief clip in a moment on that. And we're working with the Saints Foundation, who, of course, are a highly unusual organisation to be linking to from a research point of view, but they have an enormous fan base, an enormous worldwide contact on their on their internet listing and so on, and they're a highly to a population who are not always that interested in health anyway. They're perhaps more interested in going to watch matches and to have a few pints of beer. So they've been extremely supportive, and in fact some of the prizes that we have to give away later on will be for tickets, I think, with their Everton match, as well as a signed shirt, and I think it's a football too, so just keep your names in the hat. So let's just very briefly look at some of the things that have been going on. These are some nice pictures over the last year or two, not so much to pick on individuals, but just to convey the feeling of excitement and fun that these engagements, especially with young people in Southampton schools and Hampshire schools, can engender. That's a serious piece of pizza, isn't it? Hopefully some of you have had a chance to play with the amazing placenta outside. There it is being launched. This is from Physiology Friday on luckily a sunny day outside the hospital, and Lisa and others, I think, found fantastic engagement to the general public just going in and out of the building. And of course now we've got a nice new entrance to the building. It's a much nicer place to come and visit, and even a nicer place to stop and loiter and talk. And here we are at the Big Bang Science Fair in February of this year. And the New Forest Show last year. I'm not quite sure what Jane and colleagues are actually selling here. They won the best trade stand. It's somewhat puzzling, but I'm sure it's all in a good cause. And then STEM careers, Winchester Science Centre, as well as traditional kind of chalk and talk sort of teaching in Bishop Wordsworth School along with AS students. And of course later on we'll have a whole range of students joining us for our question time event. And then there is Lucy's giant Kaplunk thing here, which Kirsten, I think, is somewhat puzzled by as her little charges there. The nice thing I think about this is that nothing happens until you pull that critical thing out, which might be labelled, I don't know, exercise or something, and then the whole lot goes down, and that's life, isn't it? It's okay till it becomes catastrophic. Let's just see a very brief video of LifeLab for those of you who haven't actually been in it. This is one that we prepared earlier, as they used to say, on Blue Peter.
Welcome to LifeLab. We're based up at Southampton General Hospital. Here you will get to find out about your own health using a range of different scientific equipment, investigate DNA, meet scientists, and discover how you can reduce your own future health risks. We look forward to welcoming you to LifeLab soon.
Hi, today we have used the ultrasound machine, which is to check our arteries. Today has been really fun because we get to find out loads of facts about our bodies, which is really interesting.
Hi, today we are at LifeLab, and in this session we are learning how to carry out CPR properly. This is important because if we are ever put in an emergency situation, we will be able to carry it out properly and help the person.
Hi, we're at LifeLab today, and we're going to see 3 scientists behind these doors, and they're going to talk to us about how they use science in their work. For me, I really enjoyed learning about the other scientists. It's so unexpected that you find this in a hospital of all places, because you expect loads of people being treated, but there's actually lots of researchers going on behind the scenes as well. In this session we got to dress up like real scientists and study our own DNA. It was a lot of fun.
I think LifeLab is a really good learning opportunity for other students to take things out of the classroom and try things that we can't give you in school. This morning the students got an opportunity to try a range of activities, doing body measurements, all sorts of equipment and kit we just don't have access to. I think every single one of them I know I've spoken to has thoroughly enjoyed that activity they did this morning, and I would encourage everybody else to come and try it.
The best time of the day was definitely the experiments, as it got us all involved using all the new equipment that we'd never used before instead of just telling us what to do. I think other students should attend LifeLab because it's such a great day and it's such a great experience that you're probably never going to get unless you take up this role, and it really helps you decide whether you want to do something like this or not.
They steal the show, don't they? As those of you who have been involved in LifeLab, never worked with children or animals. I think, as one of the pupils there said, actually the Meet the Scientist component of what they come and see and do here is probably the thing that's more influential than anything else. So I do encourage you all to train to be Meet the Scientists so that you can go and meet the kids. So in wrapping up, I've got just 3 things to say really about just things to think about in taking things forward. And it seems to me that we need three different things, and this is a trial. One is we need a very clear image, because as an old person like myself is surprised to find actually young people talk to one another through images more and more. They share video clips, they share images. So we went to a lot of work in preparing today to come up with this single image. Looks like it's just three hands, but actually there are 4 or 5 different images all blended there, and it was a huge amount of work to do. I don't know whether you like it or not. I'd certainly like some feedback, because whether we retain it on the front of the IDS where it's writ large, or whether we try and modify it in some way or think of something else. But to me, it captures the passage of something from one generation to the next, the influence of a prior generation, the way that the young person's life will perhaps be altered as they grow up, and so the cycle of life through from the young mother there passing the heart as an image of life and health to the next generation. Then we need a very simple hook line. David Barker used to say, if you can't explain what you're doing to the person who works in behind the bar in the pub down the road, well then you're wasting your time. So we struggled and struggled to think of maybe just 3 words that capture what we do. 3 words, they've all got to be pretty freighted with meaning. There's no room for any waffle or persiflage there. So we came up with developing healthy lives. And then last of all, as I said at the very beginning, you need a story. You need something that gives a narrative, really, of what we're trying to do. And I learnt this from talking with Dr. Giovanni Corrado, who's a science communicator, who some of you have met, who's been very helpful in making me think through how we do this. And I think it was probably Giovanni who said, well, you know, your message is all awfully negative. You've written books like Fat, Fate and Disease. Who's going to buy that? How depressing is that? Can we not turn this over the other way and make this a positive thing? And I said, what do you mean? He said, well, maybe it's the gift. Maybe it's the idea that actually there's a fantastic opportunity. There's something that every single young person can do to pass on a future life of health and well-being and prosperity to the next generation. And actually, there's something that everybody can do, however old they are, actually, to use the gift that they've been given from previous generations, genetic, epigenetic, environmental, all the stuff we've heard today, and somehow use that to make their lives a little better. And it seemed to me that suddenly, by turning away from developmental origins of health and disease to something that's about opportunity and potential and human capital, if you like, we might perhaps have a nice story. So really that's all I wanted to say, and it's now a great pleasure to hand over to the director of our Life Course Epidemiology Unit, Professor Cyrus Cooper, and he's going to tell us about the big picture. Cyrus.
That was a very good dodging of questions. Do we need to move on? There. Oh, thank you. Well, little did I imagine 36 years ago, Mark, when I went to the Royal Southampton as an SHO in medicine, that I'd be standing up to say a few words about the Institute of Developmental Science. And my starting remark is that I'm really proud and privileged to be able to do so. And that's in large part because when we returned from Mayo in the early 90s, I had the privilege of meeting you a few years shy, as we'll see in one of my slides, off that 1997 Eric, Mick, Howard period. That joint infrastructure funding bid for the building that we wrote and spent a lot of time putting together and only got a bit of served as the sort of building block of what we've seen today that extends through the University of Southampton and provides for so many of you that are here today a unique selling point of the science that you undertake. And as there's probably only one attendee today who dates back to those days when David was actually a consultant physician, Charles, I had the rare privilege of knowing that I was looking after the wards when my consultant was attending the Clinical Pharmacology Society. And I don't know how many members of that society are left standing today, but only one is in this room that I'm aware of. So my task is to tell you a little bit about the institute within the faculty and to allude to the relationship it has with the unit that I've taken over direction of from David Barker in 2003, the Life Course Epidemiology Unit, and then the NIHR Biomedical Research Centre, whose leadership are actually in the audience today. So I'm just really iterating for you their achievements, to contextualise outside of the faculty within the university the work of the Institute, and that's principally the Institute for Life Sciences that Peter Smith was able to join us for this morning and has put together a life sciences strategy, and to talk a little bit with the closeout on the global, national and regional impact on health policy. So first of all, the University of Southampton. There are some of you who are not from here. And for you, the lift conversation description of the Faculty of Medicine at the University of Southampton is that it is small but perfectly formed. And the reason for that is that that small number of extremely focused investigators and educators work on a defined number of themes to try and achieve international success. And at a time when resources are concentrating on the triangle and on areas of sort of large-scale infrastructure capable of solving the problems of tomorrow, we pride ourselves in the areas in which we exhibit excellence on being able to deliver that with very small, well-functioning, integrated, cross-disciplinary teams. And that's why Southampton figures with an MRC unit, a CRUK centre, a BRU from the NIHR, which has become, well, two BRUs that have now become a BRC, a Clark, and an ARUK Centre of Excellence. These put it on the sort of same platform as institutions that are substantially larger. And I'll give you an example of that from my own life. Our 1,000 staff in our faculty at £100 million a year are entire fit within a university of £550 million a year to £750 million a year. That turnover, with our trust turnover of about the same amount, is dwarfed by the Division of Medical Sciences at the University of Oxford, where I also have an employment affiliation. And we achieve what we do because our institutes fit within this academic unit structure, and it's the Human Development and Health academic unit that Karen Temple leads, which has facilitated this recent step change that we've seen in the Institute of Developmental Science. So let's go to the underpinning theme. David Barker, as you've heard, showed that the ecology of death from coronary artery disease today resembled the ecology of infant mortality some 7 decades earlier. His observation at the ecological level was replicated in individuals. He went to Hertfordshire and found the birth records of people who were then traced and showed this relationship of declining birth weight being associated with increased risk of death from coronary artery disease. He proposed, some of us would say in rather rudimentary terms, a mechanism, the idea that there might be persisting changes in structure and function caused by environmental stimuli during critical periods of early development. And he articulated these hypotheses through the 90s, such that in the period spanning the Institute of Developmental Science's lifetime, they became publicly available on the front cover of magazines such as Time. And to go back to the origins of this area of science, the first terminology we adopted was fetal origins of adult disease. And I recall the mirth that was expressed when it was abbreviated to FOAD, and quite rapidly, because of the connotations which can't be repeated here, that were sort of associated with those four initials, became the developmental origins of health and disease. And this meeting was one of the earliest international workshops on fetal origins of adult disease. It was held in 1994, only about 60 yards from here in the old postgraduate medical centre. And you can see our origins with David, Alan Jackson, Geoffrey Robinson, Peter Gluckman, Catherine Law and Caroline Fall in bibs, and Keith Godfrey and myself in nappies, Clive sticking up at the back somewhere there, I think, and one or two of the rest of us left standing from those days. And it was Mark's attendance at those meetings that led to the initiation of the Centre and then the Institute of Developmental Sciences. The unit is currently divided into 5 programs. 3 of those are musculoskeletal, and 2 of them relate to adiposity and its related consequences. All of them have as their foundation the developmental contribution to later dysfunction in those 2 phenotypic areas. Our principal objectives are to move from observation to an understanding of mechanism and to the development of intervention. And you've heard in all of the presentations today the form that takes. And to clothe that with one scientific narrative, again, that's been alluded to, 12 years ago, we published the association of maternal vitamin D insufficiency with childhood bone mass. That was then completed in interventional terms with the publication by Nick Harvey and the team at the unit of the trial linking intervention on mum's vitamin D with a supplement during pregnancy to the baby's bone mass, particularly for winter births. And the mechanisms for those associations and that change are studied in the labs of the Institute. And they, in the observational data and subsequently in the interventional data, showed that alteration of methylation at a key heterodimer in the vitamin D signaling pathway, RXRA, was linked to both the bone mass of the offspring and was perturbed by the vitamin D supplement. So that's just an example, one among many, of how the observation, the mechanism, and the intervention link together. The third axis to the Institute, the Life Course Epidemiology Unit, is the Biomedical Research Centre. And that is currently led by Robert Reed and includes within it Keith Godfrey for the nutritional component and Ratko Djukanovic for the respiratory component. And I'm not going to take you through this slide other than to rehearse that these 2 large scientific theme areas are complemented by 3 cross-cutting themes: data science led by Karen, microbial science led by Rob Reed, and behavioural science led by Lucy Yardley and Mary Barker. And the nutritional component, which of course involves the Institute intimately, has an absolutely clear life course approach to it, going from preconception and adolescence through midlife to older age, as Philip so elegantly demonstrated this morning. And again, observation fuels intervention. So observational work, which you've seen, identifying pregnancy and preconceptional risk factors for childhood adiposity, matures into supplementation studies, preconception, the NIPA trial that Keith showed you, linking that to maternal insulin resistance and subsequent body form of the offspring. And the mechanisms that underpin that, Karen Lillicrop, Chris Bell, and the epigenetics team in the Institute tie up through looking at methylation across the genome. Now to move out with the faculty to the university and the new life sciences strategy developed by Peter Smith and colleagues at the Institute for Life Sciences. For us, those key areas of translational research and interdisciplinary research, particularly moving out with the biological sciences to the engineering and physical sciences and the social sciences, are core to that institute. We have 320 research group leaders across all 8 faculties represented within the institute. And half of the membership only is made up by biomedical, and the other half by the physical and social sciences contributions. Peter has 4 broad thematic areas: health and medicine, living systems, life technologies, and data insights. And you can see that the life course occupies one of the cross-cutting themes within those areas. And the Institute has a strong integration with the Institute of Developmental Science, the IDS, has a strong integration with the IFLS. So that, for example, there have been joint pilot projects supported over the last several years. There have been a number of joint PhD studentships. Peter organized an epigenetics and evolution conference some years ago. There's been all the work on stronger active bodies embodied in FortisNet that touches on the contributions that Richard gave and his team gave in regenerative medicine. And there has, of course, been the recent Renovos launch. And lastly, policy impact. Mark has shown you some of the policy impacts made by the Institute. Whether we're talking about the World Report on Aging and Health from 2015-16, the WHO Europe initiative on life course intervention to promote human health, ending childhood obesity, or our own national Department of Health research strategy and the annual report of our Chief Medical Officer, the scientific contributions of the Institute have figured throughout. So I think that I just want to stand no longer between you all and tea. I want to say a huge thank you to the organisers of today, to Lucy, Mark, Karen, and all of the team leaders whose teams have contributed. And on behalf of all of us in the rest of the Faculty of Medicine, the university, and collaborators out with, to say a big thank you to all at the Institute for Developmental Science at the University of Southampton. Join me in thanking them. Thank you very much.
Thank you, Cyrus. The bad news is I think the tea's not till 5. Is that right, Lucy? So we have a break now because we're regrouping for our evening event, Building Superhumans. So that means that developing superhumans, isn't it? Building superhumans, question mark. So that means that we've got children students from Southampton schools joining us, as well as some other people registered for the event this evening. Our panel, chaired by Professor Lord Robert Winston, who is on his way, South West Trains or South West Railways, whatever it's called, permitting. So tea will be served from 5, and then that event will actually start, hopefully bang on 6 o'clock. So thank you all for coming. I have one other duty, of course, which is to announce the winners of the first tranche of prizes. I won't say what the prizes are, because to my mind, one of them's a lot better than the others. Like one of them's a survival kit, so that's the last thing I need. And another one's a tech box. What the hell's a tech box? Anyway, they can be obtained from Jane out there on reception. And the winners are, ladies and gentlemen, Carolyn Wallace, Gado Elamin, Kath Woods Townsend, this must have been fixed, Alia Hafiz Abbas. So the 4 of you, very well done. And it was just a game of pure luck, but collect your prizes over there, and hopefully we'll see you all again to kick off at 6 o'clock.